Hi there, boys and girls. I hope your STPM 3 revision is getting on well. Today, BioWorld is presenting a brief analysis of STPM 3 past year questions. I've made this video on request from one of you named @Pirate. Though it is like 3 days away from your actual exam this Tuesday, today being Saturday, I believe nothing is ever too late. Hope that you will find some useful information from today's video. So come, join me. Now, I don't know if you watched my analysis for semester 1 exam, but if you went through it, you actually would have noticed that 78%, I actually counted, 78% of what I anticipated would be tested was tested. So because of that, I'm feeling a little bit confident now about making some suggestions for semester 3 exam. But as usual, my disclaimer, follow at your own risk. Okay, however, you can always use my information as a guide. So let's have a look at the overall analysis. Of course, there's a lot to cover. We have from the year 2013 right up to 2022, where you will also notice that there are some occasions where there is a ulangan paper, repeat paper. Once upon a time, there was repeat for semester 3, but once the ministry realized that this delayed IPTA intake, they did away with the repeat papers. Okay, so you can have a look, overall look of all the chapters, subtopics, and the years it was tested. But to make things easier, I'm going to break it down into chapters. So let's start with chapter 14 first. Chapter 14 is made out of 5 subtopics: taxonomy, diversity of organisms, biodiversity in Malaysia, threats to biodiversity, and conservation of biodiversity. Now, if we look at the breakdown of years where these questions were tested, you will see that most recently it was biodiversity in Malaysia that was tested in 2021. Okay, so when we want to sort of predict probability of questions, it is usually the questions that have not been tested in a long time. So for the longest time, conservation of biodiversity hasn't been tested, last being 2015 repeat. This is followed with diversity of organisms in 2016 and maybe threats to biodiversity. So I'm banking on these 3 subtopics to be focused on more. Let's go into depth for each of these subtopics. Okay, there was a delay in the screen just now. All right, here is the first subtopic that I am banking on, that is diversity of organisms. Not everybody's favorite topic because there's a lot of memorizing to be done. This is the syllabus item. And of course, in my playlist, I haven't done much for semester 3, and I'm so sorry for that. I only managed to do 3 simple videos, and that covering kingdoms of Protoctista, fungi, and plantae. Okay, but if we look at the questions that have been tested over the years, in 2013 repeat, there was essay questions on Cnidaria and Mollusca. Then in 2014 repeat was a structure question. This was covering Bryophyta from species Funaria. Then it came with an essay in 2015 repeat where they tested on Chordata. And in 2016, a structure again covering a few organisms, including the fungi, the Angiospermophyta, Platyhelminthes, Mollusca, and Chordata. Okay, so if we see, based on the pattern of questions, Mollusca and Chordata seem to be quite a favorite. So if you are feeling that the diversity of organisms is a little too much to recollect, maybe you can pay a bit of extra attention to Mollusca. If you remember, this is SpongeBob's pet and Gary, yeah, his name. So the characteristics, lah, okay, muscular foot, and then you have a mantle, you have tentacles, you have a radula as the mouth. So if you know about 5 characteristics, you should be able to answer the essay question. And of course, the chordata. Okay, characteristics of the chordata will be, the main thing is to have a notochord, a dorsal nerve, hollow dorsal nerve cord, pharyngeal slits, post-anal tail, and then you remember you must end your sentence with at one of the stage of development. Okay, so that's about what you prepare for this subtopic. The next subtopic, okay, that is threats, I combine with conservation, because if they are going to ask an essay, they'll definitely combine both. So we look at the syllabus. For threats to biodiversity, you must be able to explain both natural and man-made factors. But of course, most popularly, they will ask you about the man-made factors, okay, that threaten the biodiversity. And then for conservation, you need to know the various measures to conserve using in situ as well as ex situ. So if we see 2013 and 2015 repeat, basically they ask for the measures to conserve biodiversity and also to discuss in situ and ex situ. So this topic, actually, if you have a good general knowledge of your environment, you would be able to answer. So that covers chapter 14. Let's move on to chapter 15. Now, chapter 15 is divided into 6 subtopics, and here are the distribution of all the past year questions. Like 2014 here is because it was tested both as an essay as well as a structured question. So out of 6 topics, we can minimize your revision or your quick look through before your exams based on the topics that have been tested or have not been tested in a long time. So clearly you can see 2013 level of ecological organization, definitely a good probability. Then we have 2016 energy flow and 2018 carrying capacity. The others all were tested last year. Okay, so highly improbable that they will be tested again. Okay, here we look at the first subtopic. Syllabus-wise, what you need to know is mainly about the interactions. That's what was tested in the 2013 question. The interactions between the autotroph and the heterotrophs, the relationship between prey and predator, the relationship with decomposers, and then the interaction between the biotic components and the abiotic environment. Okay, so things like this normally represented in the form of a food chain. So it's quite simple to prepare for this. Just remember definition of each type of level in the ecosystem. We go to the next subtopic. Here it's about energy flow. Very high chance, in my opinion, to be tested because it was last tested in 2016. Syllabus wise, we look, you are to be able to describe the energy flow and efficiency of energy transfer in either a tropical rainforest or a lake. So in past year questions, the 2015 essay question was about energy flow, and 2016 structure question talked about energy flow using the lake ecosystem. So here's a sample of a simple food chain. So when you are going to talk about energy flow, you just must make sure that you mention that you describe the food chain, and you mention that at every level 10% is passed down the food chain, but as the chain gets longer, the amount of energy gets less. So the food chain is limited to a certain number of trophic levels. You must mention about the energy loss, okay, and that's about it. But if the question were to ask you about energy transfer, then you mention gross primary productivity, net primary productivity, and then you must mention the percentage, 10%, 20%, 90% energy loss. Okay, so I'm just trying to give a brief uh, memory recall for you. I'm not going to go into the detail of the theory, but hopefully by listening to what I'm saying, you can like jog your memory about what you have studied on that topic. Okay, moving on, we see the third subtopic for this chapter, that is carrying capacity, last tested in 2018. The syllabus only wants you to know what is the meaning of carrying capacity, sustainable development. So as usual, know your definition for both these terms and to be able to explain factors that will limit the population size. So when questions on this come out, you see twice structure has been tested on this. The carrying capacity graph, okay, reflecting the population size is what will be tested, this graph. So you should be able to identify this is the part where there is carrying capacity. Then we'll have to explain why the population begins to become constant just below the carrying capacity. So this is usually related to density dependent factors. Okay, and if say like this is a S curve, we also have populations that are J curves. Okay, so usually the J curve graphs become limited because of density independent factors. So you have to know the difference between them. Density dependent factors are dependent on the population size because then there's competition. So there will not be sufficient resources for them to survive. So that's why they have to limit their population. Whereas in the case of density independent, it's got nothing to do with the size of the population. It's just that maybe the population increased because of mating season. Then no more mating season, so the population starts to reduce. Or maybe there is a forest fire, okay, or maybe somebody sprayed some herbicide. So things like this are density independent. So that's about what you need to know for chapter 15, ecology. Let's look now at chapter 16. Selection and speciation. This is a short chapter, only 2 subtopics, natural and artificial selection as well as speciation. Now if we look at the past year questions, there's been quite a lot on the speciation part. Okay, and the selection part also has been tested in 2020, speciation in 2021. So some may say that, oh, okay, this chapter may not come out because it's been quite recently tested. But if you look at the pattern, it's been like every 2 years. Okay, if you notice this, you see 2016, then 2018, then 2020, then 2021. So I feel like it has a probability of coming out this year. And unfortunately for this chapter, I cannot see a shortcut, meaning that we actually need to know everything. So let me try to just give a summary of what is the content in chapter 16. Okay, we start with the natural and artificial selection syllabus. Okay, you are to be able to describe continuous and discontinuous variation. Then you should know the modes of natural selection, 3 of them mainly: stabilizing selection, directional selection, and disruptive selection. Okay, we also have sexual selection and polymorphism, okay, and artificial selection. But if we look at the past year questions, the topics here that were tested are in 2013, okay, modes of natural selection, 2014, modes of natural selection, and even the 2019 structure was modes of natural selection. Only in 2020, they talked about continuous variation. So meaning that under this subtopic, natural and artificial selection, I think it's safe to say that if you focus mainly on the 3 modes of natural selection, you should be able to answer the question. So over here, we have the graphs that are usually related to these modes of natural selection, okay, where you see that the red is the initial population graph, and the blue is the graph after selection. To just briefly explain this to you, let's take a population of grasshoppers. Let's say this grasshopper population has sort of a continuous variation, okay, where the color ranges from a very light cream color, green, and eventually a very dark brownish green, okay. So meaning that, let's say in our example here, originally in that ecosystem, there were these three different groups of grasshoppers living happily ever after. But let's start off with the first one here, the stabilizing selection. Okay, let's say after lots of heavy rain, the population of grass in that habitat became a lot. So this green grasshopper naturally, you see, becomes very well camouflaged, whereas the cream and brown grasshopper become extremely visible. So what's going to happen? They will become prey to predators like, say, birds. So their population will decrease, causing the graph to become more narrow. Okay, and this is what we call as a stabilizing selection. But let's say then hot season comes along and these green grass start to die. So they become all brown like this. So now you can see this kind of environment is not suitable for the cream color nor the green color grasshopper. It became more suitable for the dark brown grasshopper. Okay, or vice versa. It could also become more suitable for the cream color grasshopper. So this is what we call as a directional selection. Lastly, the disruptive selection is when there are two different environments. Let's say I have grass that is cream and grass that is brown. So this time nature will select against the green grasshopper, so their population becomes reduced. So in disruptive selection, I will have two curves where the intermediate population becomes small. So I hope this gives you a rough or a quick reminder of how natural selection occurs. Okay, do read on further to know of their consequences. Moving on, we go to speciation. The syllabus for speciation expects you to know about the process of isolation, genetic drift, hybridization, and adaptive radiation, as well as the importance of speciation. But if we look at what questions were tested over all these periods of time, most of the questions you see, starting with the structure, was focusing on isolation. Then the 2014 essay was on genetic drift. 2015 genetic drift. 2015 repeat was on isolation. 2016 genetic drift. 2018 isolation. 2020 isolation. 2021 genetic drift. Of course, I can't say 2023 what will come out, but the focus you can see is between these two topics. So for those of you who don't like the topic of speciation, rather than skipping the topic altogether, have a look at these two parts. Okay. Now, isolation, let me just help you a little bit. We go back to our grasshopper population. Okay. So let's say they all live in the same habitat, but a river develops between this population, separating them. So this we call as allopatric speciation. Okay. Now, let's say after some time the river dries up and this population is meeting one another. Now, they can become different species, okay, because of some changes that occur to them. This is called sympatric speciation. Okay, so during sympatric speciation, there may be reproductive isolation, whereby you find that maybe the population of these brownish grasshoppers are able to mate, but for some reason, due to reproductive isolation, either prezygotic mechanism or postzygotic mechanism, they are unable to mate with the other grasshopper. So these are forms of isolation. The allopatric speciation is due to geographical isolation. Okay? And sympatric speciation most likely is due to reproductive isolation. Then we also have genetic drift. Genetic drift can occur two ways. The first way is that a small population is separated from the main population. So in this case, let's say the green grasshoppers, for some reason, migrate to a new area, and over a period of time, they become two different species. So this is known as the founder effect. Or if say like an random event, such as say spraying of herbicides, cause a certain population of the grasshoppers to die. So then now we find that we have a smaller population from the original one. This is known as the bottleneck effect. So please read up on those two, isolation and genetic drift, in preparation for your exams. Now we go to chapter 17. This is one of the hugest chapter with 8 subtopics, right? And even if I look at the past year questions, there's so much, okay, that it was quite a challenge for me to type all the years. Okay, so if we look now at the distribution of questions or the likelihood of topics being tested, once again, my focus will go to the topics that have not been tested recently. For example, non-Mendelian was last tested in 2016, genetic mapping in 2014, and gene expression 2020. Okay, and actually this regulation of gene expression, one of my favorite topics, has never been tested, although in the old syllabus it was tested frequently. So I feel, who knows, this year. Okay, so let me go through each subtopic now. The first one, we combine the non-Mendelian inheritance with genetic mapping. We look at the syllabus for non-Mendelian inheritance. In non-Mendelian inheritance, we talk about cases where the genotypic and phenotypic ratios don't follow Mendel's. Mendel's for monohybrid cross is 3 to 1, and dihybrid is 9:3:1. But in the case of incomplete dominance, we will get a phenotypic ratio of 1:1. Co-dominance also will get 1:1. Multiple alleles, we will get 1:1, right? And then we also have things like lethal genes, polygenes, linked genes, sex-linked genes, epistasis, okay, and pedigree analysis also involved. So sometimes students who don't enjoy genetics, you find this such a burden, okay? But I'll tell you a little good news. All the questions that have been tested from these 5 that we have before us mainly are focusing on linked genes, as well as from genetic mapping, they are asking you to calculate the distance between the two locus of genes. Okay, of course, there was also questions with sex-linked genes. So the other questions here, oh, of course, there was one more on ABO blood group. Okay, but you see, you don't really have to know everything. If you are going to prepare everything, it is more suitable for objective. But if you're targeting an essay or a structure, my bet will go to linked genes. Okay, so just to remind you a little bit about linked genes. Linked genes is a situation where the gene A and gene B are located on the same chromosome. In the case of Mendel, he says gene A is in one set of chromosome, gene B in another set, meaning that during gamete formation, they can do independent assortment. But when they are linked genes, they cannot independently assort, meaning that gene big A and gene big B is stuck together. Gene small a and gene small b also stuck together. So when we write linked genes, we have to write AB stroke, big A, big B stroke, small a, small b. You cannot write the way you do for Mendel, that is big A, big B, small b. Okay. Now, in the case of sex-linked genes then, there's only going to be one gene. Okay, unlike linked genes, we have two genes, A and B. In sex-linked genes, you only have one gene, either big A or small a. But if you are writing for a female, you have X superscript A. It could be big A or it could be small a. However, if for the male, you have to write XY, where you only write the superscript A with the X chromosome. You don't write anything for the Y chromosome. So this is just some reminders on how to write if they ask you to draw a genetic diagram. Okay, let me move on to explain, because personally, even with my own students, I am banking on the linked genes topic to come out as an essay or even structure. So that is why I'm also sharing with you a little bit of story on this. Okay, here we have normally the diagram or the story they will tell you is a purebred was crossed and then the F1 was test crossed. So the F1 usually is a heterozygous in genes, and when you are doing a test cross, it will be homozygous recessive. So during gamete formation, what's going to happen is, since they can't independently assort, you will only get two types of gametes, mainly big A, big B, and small b. Then the other chromosomes also will form gametes, okay, that will combine during fertilization to produce offsprings with parental type genotype. I mean, they're exactly like their parents. Big A, big B, small b, or small a, small b, small a, small b. However, we find that while this happens in large numbers, so normally the data you will get of the offspring will be, the parental types will have very big numbers of ratio 1 to 1. Then you will see a smaller number, okay, also ratio 1 to 1, but smaller number. This happens due to crossing over. So during crossing over, the genes become exchanged. So now the chromosomes will have mixed gene. That is now big A, small a, big B. So now you get two new types of gametes, and then fertilization happens, and the offspring now has a different combination of gene. That is a mix now of big A, small b, or small a, big B, small a, small b. So since they are mixed, we call this the recombinant type. Okay. Then questions, of course, will ask based on genetic mapping to calculate crossing over value. To calculate crossing over value, you just have to take the number of offsprings that are of the recombinant type, divide it with the total number of offsprings, meaning that you plus the parental type as well as the recombinant type times by 100%. So you'll get your crossing over value. Okay, now we look at the other part, the gene expression part, which was last tested in 2020, and the regulation part was never tested. Okay, many students dislike this because this topic has the experiment by Biddle and Tatum. Then this part not so bad, the genetic code table, okay, and then the process of transcription and translation. Okay, but the only question that has ever been tested from this topic has come from translation. And actually, they don't ask the whole translation process. The question, which was a structure question, was mainly about the structure of the tRNA. Okay, but even if, say, it's an essay question for gene expression, then you need to sort of know your step 2, that is transcription, how we make copies of mRNA from the DNA, then how the mRNA inserts into the ribosome and becomes translated to form polypeptide. Okay, so you have to prepare for that. And my favorite, regulation of gene expression, which I feel is so simple, okay, where you just need to know how the lag operon functions. So remember, the lag operon has the promoter side, operator side, and then the structural gene side, right? And then the regulatory gene will synthesize the repressor protein. And when you don't have lactose, the repressor protein will bind to the operator region, preventing the RNA polymerase from binding to the promoter site so that no transcription and synthesis of enzyme happens. Okay, but if there is lactose, then the allolactose will bind with the active repressor, making it inactive. So the RNA polymerase can bind to the promoter and synthesis of enzyme happens. So you can look through that topic too if you want to prepare for regulation of gene expression. Moving on. Okay, now we move on to the last two chapters, chapter 18 and chapter 19. Both these chapters have to be studied together because they are linked. You cannot separate them. So altogether, there are three subtopics and there are lots of questions tested on this. Okay, like here in 2017, it's a combination of structure as well as essay. And since they are all together, we can't skip. You have to know rDNA technology to be able to explain application of biotechnology. So let's look at each topic. The rDNA technology syllabus is actually this final part where you are supposed to be able to describe steps involved in genomic and cDNA cloning. So when you're explaining the steps, you need to know about the enzymes, you need to know about the palindromic site, you need to know about the plasmid. So all these things you have to know to understand the cloning process. So if we look at the questions that have been tested also, you see 2013 repeat was cloning, 2014 was cloning, 2014 repeat was cloning. Then 2015 was about the libraries, 2017 about the enzymes. Okay, but as you see the years arranging themselves, you will notice that the majority of the questions came from the cloning part. So you have to know the cloning process. Let me briefly explain the cloning process to you. Okay, cloning has mainly 5 steps here: isolation, insertion and ligation, transformation, screening, and finally amplification. Okay, so it starts with the isolation of a plasmid and donor DNA. And during the isolation process, we will have to use restriction endonuclease to cut the plasmid as well as the donor DNAs so that they have what we call as sticky ends. Then what we do is we let the DNA fragment, target DNA, bind to the cut plasmid and then form an R plasmid using DNA ligase. Then we transform this rDNA into the bacteria. And finally, we screen the transformants, that is, bacteria that have taken up the R plasmid by growing them in X-gal ampicillin medium. So over here, you can see this is what we call as the bacteria colonies. You have the white colony and the blue colony. The white colonies are the cells which have taken up the rDNA, whereas the blue colonies are the ones that have taken up normal plasmid. Okay, so the details of this, you can go through your notes. This is just like a refresher for you to know how to explain the steps. So this is chapter 18. We move on to chapter 19. Now in chapter 19, you are going to apply our knowledge of rDNA technology. We apply it in the fields of in the field of food and beverage, agriculture, medicine, forensic, as well as public health. Now we look at the questions that have been tested. 2013 wants you to know the definition of biotechnology, whereas the other years you see, right, gene therapy was tested in 2013 as well as 2016. Genetic screening, that means amniocentesis, chorionic villus sampling, that was tested in 2014 and 2015. DNA fingerprinting was tested in 2015 repeat, 2018 as well as 2020. The structure was the one where they have the DNA bands for you to identify the criminal. Okay, the others are assays about paternity cases, how to identify who is the parent. Then in 2016, we had application in agriculture and medicine. And then creating herbicide-resistant plants was tested in 17, 19, and 20. So you can see, now as you notice, in the application of rDNA technology, questions are related to DNA technology in the field of medicine, where we use bacteria to synthesize medicine, or in the field of gene therapy, where we try to insert healthy genes into patients who have single gene disorders, or create transgenic plants. Now, the process of cloning is similar, but there are certain things that we have to state differently, which I want to share with you. For example, if you want to genetically modify the bacteria for synthesis of medicine like insulin or human growth hormone, okay, what we do is called transformation, where we isolate the plasmid from E. coli, insert the gene of interest, and then transform the plasmid into the E. coli. So now E. coli will be able to synthesize insulin or human growth hormone. Okay, but now if say we want to directly treat humans, especially those with single gene disorders. So this process we call as gene therapy, and the method to do is called transfection. Okay, so what we do is we don't need a plasmid, we just have the gene of interest. We mix the gene of interest to a vesicle that is surrounded by phospholipid called a liposome. And then we take this liposome and mix it with the patient's cell so that the phospholipid bilayers fuse and the DNA is inserted into the nucleus. So this way, this cell will be able to synthesize the necessary protein to help reduce the effect of the disease. Okay, now in the case of plants, it's a bit different. Okay, when we want to create transgenic plants, maybe with resistance to herbicide, it is different because plant cells have a cell wall. So because of that, we use a method called infection, where we use the plasmid from a bacteria that naturally infects plant cells. So that plasmid is called plasmid Ti, and the bacteria is called Agrobacterium tumefaciens. So this Agrobacterium will be able to be transformed, okay, with the plasmid Ti, the R plasmid Ti, and this agrobacterium now can go on to infect the plant cells, and in that way insert the DNA into the plant cell. So that is like a summary of how a concept of cloning is used in application. So with that, I end my extremely long video. I've never done a video this long, but I hope it has been of a last minute help for all of you sitting for your STPM 2023. Best wishes from BioWorld. May all of you achieve your dreams to enter university. Bye-bye.